Okay, we'll get started here. Hopefully you had a wonderful weekend. I was able to go to my brother's house in Orange County. His son is just got accepted to a four-year college after going 2 years to a community college. So he's very excited. So we had a little bit of a celebration for him. And then I was working on this book that I'm trying to get out. Claudia knows all about this bioinformatics book, which is I've been working on for years. Hopefully I will publish it before I die. But anyway, so today we'll be talking about digestion and transport of fats. It's an appropriate follow-up to our discussion yesterday about causes of overweightness and obesity in the United States. I'll be talking about lipoproteins, the transport of fatty acids inside the cell for something called beta oxidation, and then we'll go into the mechanism of the beta oxidation pathway. And finally, if we have time, I'll talk about the regulation of fatty acid release from adipocytes. So we'll try to accomplish all that. I wanted to just let you know that on Friday, June 3rd, so not this coming Friday, but next Friday, I'll be giving a talk on some of the research in my laboratory on cancer. So if you are so interested, that'll be Friday, June 3rd, at the Annenberg Science Complex B lecture hall room. So it's a big lecture hall. I don't remember the room number of that. Does anybody remember the room number of that?
132.
Is it 132? Thank you. So room 132. It's the big lecture hall. So hopefully I'll try to make it interesting for you and talk about some of the cutting edge research, not only in my laboratory, but in some of these big labs across the United States. I think it should be, I'll try to make it as interesting as possible. So we're going to talk about fatty acid catabolism, right? So fatty acid catabolism. And fatty acids represent a principal form of energy storage, as I'm sure you are aware of. What other molecules in the body can we get energy from besides fatty acids? Anybody know? Very good, carbohydrates, right? And there's one other source that we sometimes use as well, protein. So those are really the three sources. And it turns out that if we consider on a per mass basis, it turns out that we get a lot more energy out of fatty acids than we do from proteins and carbs. So as a matter of fact, for fatty acids, which I'll just abbreviate as FAs. Okay, so FAs, we get about 37 kilojoules per gram of energy out of the fatty acids. On the other hand, for carbs and proteins, we get about 16 to 17 kilojoules per gram. So what is that? That's a little bit more than twice as much out of fatty acids. And so can anybody think of some molecular reasons as to why we get so much more out of fatty acids? Wonderful answer, exactly right. Compared to carbohydrates and proteins, fatty acids are more reduced, right? And you guys already appreciate the fact that it's the electrons that we strip from these molecules which are important for deriving energy, right? So if we look at a typical fatty acid, which looks like that, okay? That's a fatty acid. You can see that you have a lot of hydrocarbons here, and if you calculate the oxidation number for each one of these carbons, it's going to be -2, right? So -2 oxidation number, And for this last one here, will be -3. The oxidation number for the carboxylic acid will be pretty oxidized, but it is the reducing power in these carbons that's going to be important for how we get the energy from them. On the other hand, as you guys know, glucose looks like this. Okay, and if you calculate the oxidation state of those carbons and divide it by the total number of carbons out there, you'll find that the oxidation state here is going to be approximately, I don't remember exactly what, it's going to be approximately about 0, okay, on a per carbon basis. So we don't get very much energy out of glucose compared to the fatty acids. So fatty acids, we get a lot of energy out there. The other thing great about fatty acids is that they store in a pretty packed area. In other words, you don't need water to solubilize the fats, okay, the fatty acids. The fatty acids will stay accumulated together. And does anyone remember the forces responsible for allowing the fatty acids to stay stuck together? Beautiful. Van der Waals is one, and there's another one that's a little bit harder to explain. Anybody remember? Hydrophobic. Very good. Hydrophobic driven forces, entropy, right? So hydrophobic, what do we call that? Hydrophobic forces. Let's say hydrophobic forces. Basically, you want to exclude water to increase chaos or increase entropy. Okay, so those two forces drive these fats together so that they exclude water, so they don't take up very much room. So on a per space basis, the fats are really good at storing energy in a smaller space. On the other hand, as you can see here, glucose and carbohydrates, you're going to need some water to solubilize that. It takes a little bit more room. It takes some more room in order to keep the glucose and keep the carbohydrates dissolved in the cytoplasm. Very good, very good. Let's talk about how fatty acids are stored in adipocytes. So they're stored in something called triacylglycerols. And the basic structure of triacylglycerols is this. I guess this could be a Y. Okay. So that's a triacylglycerol, and it makes a lot of sense, right? This name, tri, meaning 3, acyl groups, you have 3 acyl groups, and they're attached to glycerol. So this is the glycerol moiety right there. And so this name makes a lot of sense. Sometimes we abbreviate this as TAGs, so triacylglycerols. Okay. X, Y, and Z are usually even numbers, and they could be the same or they could be different. So X, Y, and Z are usually even numbers. And the reason for that, we'll talk about hopefully later on in this lecture, the reason why they're even numbers. So typically we have an even number of carbons in the fatty acid part of these triacylglycerols. So where do we get, let's just go through an overview of what happens when these things break down. So this triacylglycerols will break down. I'm going to abbreviate this part as R1, R2, R3, just to make it easier to write this out. So what happens when you get an appropriate signal in order to break these down? What happens is you get water molecules to come in, and you break these down into glycerol. And you find sometimes this glycerol in some very fancy soaps, okay? You'll find a glycerol component. And then plus, you're going to get three what we call fatty acids. And of course, they're called fatty acids because they have carboxylic acids on them. And at pH 7, those will be deprotonated, right? So those are going to be deprotonated. So what happens then is that this glycerol then can be used for gluconeogenesis or for glycolysis, okay? And the three fatty acids, I'll write that down here, glycolysis or gluconeogenesis. And we talked about that last time, how this will enter the pathway at glyceraldehyde 3-phosphate after it gets converted. And then this can undergo a process, these three fatty acids undergo a process called beta oxidation. And in that process, you get a lot of FADH2s as well as NADHs. And this will get broken down into our familiar friend, and does anybody remember the name of this guy right here? Beautiful, acetyl-CoA. Acetyl-CoA, very good. And then this acetyl-CoA enters what pathway that we've already talked about? Citric acid cycle. Very good. Okay. Or the Krebs cycle. Okay. I'll just say CAC for citric acid cycle. Okay. And in that citric acid cycle, we're able to have a number of products. Anybody remember the products that come out of the citric acid cycle? Excuse me?
I think 6 NADHs.
Okay, good. Yeah, you're going to get any. Let's just say NADHs. Okay. Very good. GTP. CO2. Good. And FADH2. Very wonderful. FADH2. Good. And of course, a couple of these will go on and enter something called the electron transport chain. And in the electron transport chain, you'll have oxygen, which gets converted into what?
Water.
Beautiful, water. Okay. Okay. And of course, you're going to have the pumping of protons across the membrane, the mitochondrial inner membrane, and ultimately you will be able to go from ADP, an inorganic phosphate, to ATP. Okay, so that's sort of a broad overview of this fatty acid oxidation process, and it kind of reviews also what we've been talking about for the course. It's a nice diagram to kind of understand. Okay, so let's back up a little bit and look at an overview in terms of what happens when we go to the food court and order ourselves one of those wonderful $6 burgers. Okay. Actually, I was on a non meat diet, so, and I did this I think like 3 or 4 years ago, and I had a New Year's resolution starting January 1st, I think like 2013, that I wasn't going to have any meat for the whole year. And then in March, I specifically remember this, in March, I was like passing through the food court, and that Carl's Jr. just like tripped me up. And the next thing I knew, my legs, I had like an out of body experience. My legs just kind of like got in line there. And before I knew it, I was chomping on one of those $6 burgers. So that was the last time I tried to do that. All right. So what happens here when we when we eat these things? So let's take a look at your book. I'll try to keep this recording device close so you can hear it. So we swallow the $6 burger, and of course the hamburger part especially has a lot of triacylglycerols in it. Okay, a lot of TAGs in the hamburger part. So when we eat that, that goes through our stomach here. Okay, so this is on page 711. So it goes through our stomach, and when it gets through the stomach and enters into something called the small intestine, then there's a secretion of what are called bile salts. Bile salts. And these bile salts are actually created from cholesterol. So cholesterol is the precursor to these bile salts. And they're called bile salts because they're amphipathic. Remember what that word means? What does amphipathic means? Not multiple charges, but it's close. It has something to do with charge. Amphipathic. Both polar and nonpolar. Very good, very good. And it can have charge, or it could just be polar groups, but it has a polar side and a nonpolar side, okay? And that's important for these bile salts, because what these bile salts do is that they start to break up the globules of fat that are passing through our small intestine. And those bile salts are actually secreted from something called the gallbladder. And so this little green area right here is the gallbladder, and the bile salts are actually made by the liver. The liver, man, that makes a lot of stuff. Okay, so the liver actually makes these bile salts and stores it in these in the gallbladder, and the gallbladder secretes these bile salts into the small intestine in order to break apart these large globules of fat, TAGs, triacylglycerols. And basically they're called emulsifiers, and emulsifiers are exactly that. They will break down fat into smaller portions. This is a non-enzymatic process, guys. There's no chemical bonds that are being broken in this process. Okay? And you can sort of see here at the bottom a really nice picture of one of these bile salts. So here you have cholic acid. There's several different ones. And what I really like about this picture, it actually shows you the 3D structure of the bile salts. And so you can see you have the hydrophilic phase with all these oxygens. Whenever you see red, that means an oxygen. So you have all these oxygens here. Okay? And then the hydrophobic phase is this other part right here, which is in green, I guess. And so what happens is that these bile salts get together with these fat globules, and they break them down into smaller chunks. So here you have the triacylglycerols kind of in the middle here, and then you have the bile salts with the polar parts on the outside that are interacting with water, okay? And the hydrophobic part, of course, interacting with the triacylglycerols, because the triacylglycerols are very, very hydrophobic. And then what happens is that the pancreas secretes these enzymes called lipases. And if you recall my diagram that I just drew out for you, one of the first chemical reaction that occurs in terms of breaking bonds is hydrolysis. So hydrolysis of the acyl bonds in the triacylglycerols in order to produce glycerol and 3 fatty acids. So that occurs by the pancreatic lipase. Anybody want to guess what class of enzymes the pancreatic lipase belongs to? What class of enzymes? All the way back to the beginning of the course. Some people are pointing at each other. Yeah. What do you guys think? Hydrolases. Very good. So these are hydrolases. Excellent. Yes, so hydrolases work on these things. Actually, most of the enzymes that work on digestive material, so that stuff going through our digestive tract, are going to be hydrolases. They could be proteases to break apart meat, okay? Lipases to break apart lipids. What do they call those? I guess amylase, in order to break apart carbohydrates, okay? So all of these are going to be hydrolases inside the digestive tract. All right, so we've got these hydrolases working in the form of these lipases. And then what happens is that these fatty acids now you've broken them down to fatty acids, these fatty acids get removed from the small intestine and the large intestine, and eventually they get taken up by the lining of the intestine, okay? And they get through that lining, and then they're assembled into a lipoprotein, one type of lipoprotein. And the type of lipoprotein they get assembled into are called chylomicrons. Chylomicrons. I'll go ahead and write that word down here. Helps if I spell it right. There we go. Chylomicrons. And that is a lipoprotein. And they get reassembled in the chylomicrons as triacylglycerols again. Okay, so they get taken apart in order to get across the membrane and to get across those cells that line the small intestine, and then they get reassembled in our bloodstream as chylomicrons. And as I said, chylomicrons is a form of lipoprotein. Lipoprotein. So that brings us to this topic of lipoproteins. So let's pause right here and think about these lipoproteins. These are protein lipid particles that float through our system, our blood system as well as our lymph system. Okay? And they're classed based on their density. Okay? And chylomicrons are the lowest density lipoproteins that are out there. Okay? They're very, very low density. And the density depends on the protein to lipid ratio, So you have chylomicrons, you've got very low density lipoproteins, you've got, I think you got intermediate lipoproteins, you've got, intermediate density lipoproteins, you've got low density lipoproteins, you have high density lipoproteins. You have all these different lipoproteins that range from high density lipoproteins all the way to chylomicrons. The density, think about this, the density depends on the lipid to protein ratio. So my question to you is, chylomicrons, I said, has the lowest density. So would it have the highest lipid to protein ratio or the lowest lipid to protein ratio? The highest. Very good. How'd you figure that out so quickly?
Because you said it has the lowest density?
It has the lowest density.
I just figured, like, if, I don't know, I can't, like...
Something about lipids. Lowest density is going to have the highest lipid to protein ratio. Anyone want to give a rationale? If you were to calculate the density of lipids, it's going to be less than 1. Okay? And if you calculate the density of proteins, it's about, it's actually a little bit more than 1. Okay? So here's on page 712 of your book, you have this arrangement here, and we've got density of the chylomicron, and here it's less than 0.95 grams per ml. Okay? And as you go over here to high density lipoproteins, you can see that the density is 1.063 to 1.2 grams per ml. Okay? And you guys know that water is 1 gram per ml, right? So that's water. That's like our reference point here. And it's true, the chylomicron has a very, very high ratio of lipid. So if you think about salad dressing, right? The lipids kind of float to the top, right? Because they're low density. Okay? So it has a lot, so it has a lot of lipids compared to proteins. Okay. So that's how we class these LDLs and HDLs. So when you guys go, maybe you guys don't, but maybe your parents or your grandparents go and get a lipid panel from their doctor, they're basically looking at the density of these lipoproteins. Okay, so I digressed a little bit, but these chylomicrons then, they get created once they get created. And what do they actually look like? So here is kind of this is a nice picture. It's pretty simple, but it actually tells us what's happening here. So here is a kind of the structure of these lipoproteins. So a chylomicron is going to have phospholipids here. So it's got phospholipids on the outside. It's got proteins. So these are the proteins that associate with that are part of the chylomicron. And then you have triacylglycerols inside. That makes sense. The triacylglycerols are very hydrophobic, right? They're not going to want to interact. They're not going to be on the surface. Okay? So the triacylglycerols are going to be on the inside. And then you have this kind of funny structure here, and those are cholesterols. Okay? And so the polar part of the cholesterol is going to be out here. And then you have something called cholesterol esters, which are more hydrophobic, and those cholesterol esters are on the inside. So these lipoproteins, they not only carry around TAGs, but they also carry around cholesterols. We're going to talk about cholesterols in another lecture. Okay, I assure you, we will talk about cholesterol. That's a big medical topic. But right now, I'm going to concentrate on the TAGs. So let's keep our story on the TAGs. So these TAGs then, triacylglycerols, eventually they, in the form of these chylomicrons, they get transported. So I'm on page now 716. Okay, and here you have the intestine. Here you have the chylomicrons here. By the way, the chylomicrons can also assemble in the lymphatic system as well. But let's just keep it simple, talk about the blood system. So these chylomicrons, they float around the blood, and then eventually those TAGs get hydrolyzed. Okay, so they get hydrolyzed here, and the glycerol gets returned to the liver. Okay, and the fatty acids, they get resynthesized into triacylglycerols inside the adipose tissue, so inside our fat. Okay, so that's where that $6 burger TAGs are going to be residing in our fat cells. Okay, and then late at night, when I haven't, when you've exhausted all of your glucose stores, then you start to have this transport system. And this transport system is where these fatty acids now come out of the adipose tissue. And there's a protein called serum albumin, has a molecular mass of about 70 kilodaltons. Okay? And these fatty acids attach to the serum albumin, and then they go to, let's just say, the muscle, okay, peripheral tissue, let's just say the muscle, okay, or the heart. They go to the heart, okay, and once they go to the heart, the fatty acids then get placed into these muscle cells or in the heart, and they undergo something called beta oxidation. Okay, and so beta oxidation, and it's from that beta oxidation, which I just showed you in my drawing, that these peripheral tissues can have the energy to do what they do. Not only muscle, kidney, liver, other tissues as well. Okay. All right. What happens to the remnants of the chylomicrons, the part that didn't get placed into the fats, into the fat cells or the adipose tissues? They get returned to the liver. So that would be the proteins that are on the outside of the chylomicrons, as well as the cholesterol that is also on the outside. That gets returned to the liver, and the liver is sort of this big controller of cholesterol. Okay, the liver controls lots of things. Okay, this is a very complicated diagram, so I'm going to leave this diagram. When we talk about cholesterol, we'll come back to this diagram. But I just want to give you just the scoop from fatty acids. Okay, so dietary fat that you ingest is going to go through this pathway. Okay, so now we're ready to talk about what happens when these fatty acids get into these peripheral tissues. Now we got to start breaking. We have to start breaking down those fatty acids. And the first thing that happens is it enters into the cytoplasm. So fatty acids inside the peripheral tissue. Okay. And the first thing that's going to happen is it's going to get transported. Actually, if it's greater than or equal to 14 carbons, it gets transported into mitochondria. Make sure about that. Yeah, gets transported into mitochondria via something called the carnitine transport system. Okay. Let's talk about this carnitine transport system. Oh, darn it. Let's see here. I think I need to talk about... Actually, let me back off a little bit and talk about how this transport occurs. So I'm on page 725 of your book. Okay, so now we're talking about fatty acids here. And the first thing we need to do is activate the hydroxyl group on the end of the fatty acid. So the hydroxyl group that's part of the carboxylic acid has to be activated in order for us to begin to break this down. And we have to make a good leaving group. So you organic chemists, we have to make a good leaving group for that oxygen so that it can leave from that carboxylic acid. And the way we make it a good leaving group is we attach AMP to it. Okay, so we're going to attach AMP. So ATP gets hydrolyzed, and the AMP stays on the. Actually, I guess hydrolyzed is not the right word. It leaves an AMP on the fatty acid, and pyrophosphate is going to be leaving here. So you're going to have pyrophosphate leaving. Once you have a good leaving group on the terminal oxygen there, then coenzyme A is going to do a nucleophilic attack, and it will attach to the fatty acyl group, and then now the AMP is going to leave, and now that we have coenzyme A attached, the fatty acid is ready to be transported. We call that fatty acyl-CoA. Fatty acyl-CoA is going to get together with a molecule known as carnitine. Carnitine is going to transport the fatty acyl group across this inner mitochondrial membrane. So the inner mitochondrial membrane is pretty tight. As you know, protons can't even get through here, but this fatty acyl carnitine can through this transport system. Okay? And then eventually, this fatty acyl-CoA is going to get broken down in a process known as beta oxidation into acetyl-CoA. Okay, let's get started on the mechanism. Okay, so here on the right-hand side, you have a pretty good diagram here. So we have the fatty acid. They have an abbreviated form of this fatty acid. You can imagine this is a long hydrophobic chain here. Okay, but here's fatty acid, and it's going to perform a nucleophilic attack on this alpha phosphorus atom on the ATP. This leaving group here is going to be composed of two phosphorus groups, the beta phosphorus group and the gamma phosphorus, the beta phosphate and the gamma phosphate. And so pyrophosphate is going to leave. Okay? And you have this process where water is going to break apart that pyrophosphate in order to make two inorganic phosphates. What's happening here is we're driving this reaction forward by removing one of the products. And as we remove that product, we're going to make our free energy of activation change, our free energy change more negative because we're removing that product. You guys remember the equation, right? Products are always in the numerator for that equation. So we're removing that product, we're lowering that product, and so we're driving this reaction forward. Okay? In the next step, we have coenzyme A that comes along, performs a nucleophilic attack on this carbonyl carbon, and AMP will lead. This is very clear mechanisms here. So now we have fatty acyl-CoA. Fatty acyl-CoA is going to have a hard time getting across the mitochondrial inner membrane, especially if it's very long with 14 carbons in it or higher, which a lot of the fatty acids are 14 carbons or higher. So we're going to have to get across this mitochondrial inner membrane. So this is an overview of what happens here. You have fatty acyl-CoA. It's going to attach to this, I guess this is called carnitine, what is this, CPT, some kind of transport system here. And then it's going to get the fatty acyl carnitine across the intramembrane space. This is in the outer, this is the outer membrane, and then here's the inner membrane. In the inner membrane you have CPT2 get across. So let's just look at the mechanism, it might be easier. So here you have the fatty acyl-CoA. Here you have carnitine, and the attachment is going to be on this central carbon here. So this carbon right here is going to be where the attachment is going to take place on this hydroxyl group. So I have a base that's going to remove the proton from this alcohol, and that's going to perform a nucleophilic attack onto this fatty acyl-CoA, and you're going to create fatty acyl carnitine, and you'll have free coenzyme A. So this fatty acyl carnitine now is going to get across this part, and in order for the fatty acyl-CoA to be recreated, you have coenzyme A on the inside here. This coenzyme A on the inside is going to just do the opposite. It's going to reverse this, and you're going to create carnitine coming off. You're going to create fatty acyl-CoA in the matrix of the mitochondria. Okay, now that we're in the correct location for beta oxidation to occur, I'm going to talk about a classic experiment before we get into the nitty-gritty here. A classic experiment done by Franz Knoop. Franz Knoop in Germany in the early 1900s. And this is a brilliant experiment where what he did is that he took, this is 1904, I'm going to say Franz Knoop, 1904, and he discovered beta oxidation. And the way he discovered it was quite ingenious, a bit of deductive logic. What he did is that he used dogs. In those days, you could easily use dogs as your laboratory experiments, for your laboratory experiments. Nowadays, that's much more difficult, okay? Just because of the awareness of pets and everything. But in those days, dogs were used quite a bit. And so what he did is that he fed dogs labeled fatty acids. Now this is back in 1904, before the discovery of radioactivity. So he wasn't using radioactivity here. He was using phenyl groups in order to label these fatty acids. So let me just give you a quick example. He fed these dogs fatty acids that he created in the laboratory. So these are artificial fatty acids. Okay. And in this case, when he fed the fatty, and what's the label part? This is the label that he put on. So this is something that you could detect using a spectrophotometer, okay? Because it's an aromatic ring. Okay, so he used this, and he fed these dogs these reagents, and then he would check out their urine to see what they were secreting, okay, after they broke down the fatty acids. And this is called phenylpropionate. phenyl propionate. Okay, it's a good name. We can see the phenyl group, and whenever you have a propyl or propyl, that stands for what in organic chemistry? When you have propyl groups, yeah, how many carbons? Three, always three, right? Okay, so propionate. So you got 3 carbons here. Okay, propionate. Okay, and when he fed the dogs this and allowed them to digest this this fatty acid, he found. Sorry, let me go ahead and widen this up. He found that they secreted this, which is called benzoate, okay, in their urine. Okay. Now, then he fed the dogs another long chain fatty acids. These aren't that long, but he fed them another fatty acid that he created in the laboratory. Those Germans, they were really good around this time in terms of chemistry. As a matter of fact, when I was at UCLA, there was a requirement for me to learn German. Luckily, they dropped that requirement after 1 year. I was there, so I didn't have to do that. Thank goodness. I wasn't there in the early 1900s, though, guys. Okay. Just FYI. Okay, and when he fed them phenyl butyrate, he got phenyl acetate out of this. So this is called phenyl acetate. It makes sense. You have a phenyl group and you have an acetate here. So what's going on? Oh, when he saw this result, and he saw this whenever he saw benzoate whenever he fed the dogs what are called odd-numbered fatty acids. This is an odd number, that's 3. Okay. He always got benzoate. So if he had 3 carbons here, if he had 5 carbons here, if he had 7 carbons here, he got benzoate. Whenever he fed the dogs even-numbered fatty acids, so here we have 4, okay, but it could be 6, 8, 10, or 12, he always got phenyl acetate. And he wondered, well, what's going on here? Why is it that we're getting different products depending on if I start off with even or odd? Okay, and he kind of reasoned this out. Well, he said if the fatty acids were being broken down 1 carbon at a time, then the final result should always be benzoate. It wouldn't matter if it was an even or odd chain, odd-numbered carbons in the fatty acid. But because I'm getting this result, it looks like there's something called beta oxidation occurring. So let's review what that means. This is the alpha group right there. That's the alpha. Okay. I guess I'll just specially highlight this one. There we go. And this is the beta carbon right there. Okay. Now, in this one, the beta carbon is right here. So that's the beta carbon right there. So always remember, the alpha is attached to the carboxylic acid, and the beta is going to be the one attached to the alpha. And so what he was saying then is that there's something called beta oxidation occurring. In other words, this beta is the same as that one. Okay, I'll just put quotes because it's not really beta anymore, but it's that same carbon. Okay, and this beta right here is this one right here. Okay, I'll put quotes around that because it's not really beta. And he called it oxidation because obviously this is oxidized compared to this, right? So beta oxidation. And it was a beautiful experiment because it was deductive logic, number one. And then number two, it was the first time that someone used a label to trace out what was happening with these molecules. And of course, later on, they started to use radioactivity instead of these big bulky groups to follow along in these cases. So we're now ready to talk about beta oxidation in terms of the actual chemistry. So remember, beta oxidation occurs in what compartment of the cell? No, not the cytoplasm. Remember we did all this transport that I was talking about? Mitochondria. What part of the mitochondria? Matrix. The matrix of the mitochondria. Exactly. So that matrix, man, there's a lot of stuff going on there. Okay, so here we have a typical fatty acid. It's a C16 acyl-CoA. This is on page 727 of your book. They've had this thing labeled for you, where the alpha carbon is, the beta carbon is, and the first step is something called dehydrogenation, where we're going to remove two hydrogens from one hydrogen from the alpha and one hydrogen from the beta carbon. And we use FAD. Okay? So remember, FAD has that business end, okay, that allows for hydrogen atoms to be able to attach to it. And these hydrogen atoms, once they attach to the FAD becomes reduced to FADH2, and then eventually it's going to donate its electrons to the inner mitochondrial membrane at specifically the point where Q is. And remember I talked about how important Q is? Q is like this focal point where a lot of electrons get fed into it. It's part of the inner membrane of the mitochondria. And so here is another example where Q is being reduced. This is the oxidized form of Q, coenzyme Q. This is the reduced form. It's being reduced by this enzyme that sits on the mitochondrial inner membrane and reduces the Q to Q2, QH2. This is not complex 2, but it's very similar to complex 2. Very similar to complex 2. Okay. By the way, what class of reaction is going on between here and here? What class of enzyme? What class of enzyme undergoes this dehydrogenation reaction? Oxidoreductase, exactly. We are starting to strip the fatty acid of its electrons in order to create a proton motive force, in order to create ATP, right? We're gathering all those electrons. Okay, now we have a more oxidized fatty acid, but we're not done here. We're going to get more from this, okay? The next step is something called hydration. So we get hydration occurring. And then next we get, so now we have an alcohol on the beta carbon. So remember, when hydration occurs here, we're going to get the H2O is going to perform a nucleophilic attack on this beta carbon, because it's the beta carbon that's going to get oxidized. Beta oxidation, right? So you know that eventually that carbon, the beta carbon, is going to get entirely oxidized to carboxylic acid. So don't try to attack the alpha carbon. Always try to attack the beta carbon, okay, with the water. And so now we've got an alcohol here, okay, but there's been no change in oxidation at this point, okay. And now we have NAD plus going to NADH. Therefore, we're going to have a hydride transfer occurring. So a hydride transfer is going to occur, and we're going to create NADH from this. There's been a mistake here. I want to point out a mistake. Okay, and the mistake is they have labeled this hydrogen red, and it's not. It shouldn't be red actually. It should. The hydrogen, the hydride actually comes from this this hydrogen right here. Okay, so a little bit of a mistake. Oh, wait a second. Hold on. Let me make sure that's right. Oh, I'm wrong. Okay, nope. The book is right. When I think about this, it is correct. The base is going to come along, take off that proton, and then this hydride will move and attack this. Yeah, no, your book is right. It's correct here. The hydrogen does come from the beta carbon. We're going to stop right here, at this point right here, and we will go on and continue beta oxidation on Wednesday. Don't forget, Friday, there's no class. I will put up a video lecture for you.